Okay, I think we have numbers setting up, so let me start. So hello everyone, thanks for joining my talk. My name is Ellie Cho and I am an application specialist for image analysis at the Biological Optical Microscopy Platform at the University of Melbourne. So this is as a part of our Fundamentals of Fluorescence Microscopy series. So most of you probably have attended lecture 1 and 2 from Paul and Gabriella yesterday and the day before. And today I'm going to give you an introduction to bioimage analysis. So if you have any questions during the talk, feel free to type your questions in Q&A and we will go through them at the end of the talk. And also the recordings of this talk will be available on our websites after this, so you can check it out if you have missed any part of this talk. So let me start. Okay. So in this talk, I will first talk about what needs to be considered in planning bioimage analysis. Then I will go through the basics of 4 steps of image analysis workflows, which is acquisition, preprocessing, object detection, and measurements. Then I will share some example analysis workflow, followed by discussing the last step of the workflow, which is reporting. So the goal of bioimage analysis is to obtain quantifiable information from the microscope images of biological samples. So for example, in these 2 fluorescence microscopy images, we can visually tell that the cells are shown on the control group and the treated group looks different. But then how can we describe this difference? Perhaps we can say that the cells in the treated group look slightly rounder to start with, but how can we say how much they are rounder? In other words, how can we quantify this difference? One of the useful quantifier for that will be measuring the width and length of each cell and present as width to length ratio. So this would be closer to value of 1 as the cells are rounder and closer to 0 if it's elongated. And once we measure for each cells, we can then plot the value and run statistics. So this number crunching process can be simple and straightforward if we have ideal microscope images that is already optimized for it. But in many cases, we are seeing researchers trying to analyze images that are not really suitable for certain quantifications. I'm just going to stop video because that's on my presentation. So okay. So, and in fact, if you're planning to analyze your microscope images, this analysis part will be the last step of the entire workflow. And the first step is to plan your quantifications. That's to determine what needs to be measured, of course. And then we can determine the suitable microscope techniques and then prepare the sample based on those plans. So the planning should be taken place properly before any measurement begins. So let me give you an example. Let's say we want to measure the colocalization of 2 proteins in mitochondria. So because the target structure is very small and also because we want to measure the colocalizations, we will need a super resolution microscopy to get the best resolution possible. Then to be suitable for super resolution microscopy, we can prepare a monolayer cell culture on a cover slip or cover slip in chamber slides rather than thick bottom culture plates. And since we're going to measure the degree of colocalizations, we will also need to think about having a negative control sample where we're expecting to see no colocalizations, and also the positive control sample where we're expecting to see high degree of colocalizations. And once all of these plannings and preparations done, then we can run the imaging experiments. and measuring the colocalizations will be relatively straightforward. Well, now let's consider a slightly different scenario where we did not use the correct instrument. For example, if we have used some microscope that happens to be available near our lab and that we can only take low resolution images, then our images will not have sufficient resolutions and we may get the very wrong representation of our sample. And this will lead to incorrect colocalization measurements and the result may be wrong. So sometimes we may not know what kind of feature we need to measure because the actual biology may be unknown. And even if we know what we need to measure, the observation may tell a different story. So we need to carry out a pilot study always before undertaking a full-scale experiment. And from the pilot experiments, you will be able to optimize settings and readjust your plans. And once optimized, we can then run full-scale experiments. And if you're not sure what's possible or don't know where to start, it is also a good idea to contact your local core facility staff, or you can contact us at the planning stage. Okay, now let me get to the second part of this talk, which is introducing the general workflow of bioimage analysis process. Let's say we have acquired a raw microscopy images like this. And generally, image analysis workflow involves pre-processing of raw microscopy images because we might have to correct any defects happening during acquisition, and segmenting objects or defining region of interest. And then we can measure many different type of quantifiers and report those results. Well, depending on the type of analysis, we can either use a single software package that covers the entire workflow, which means all of these steps, pre-processing to analysis to reportings, or we may have to use different image analysis softwares for each steps because we might need to access specific functions. So I'll go through these each steps in more details in the following slides. And also there are many image analysis programs available in the world, so finding the best tool can be quite challenging. But luckily there's some search engines available, which is developed by the Network of European Bioimage Analysts, NUBIAS. And in these websites, we can get curated information of image analysis softwares and which types they are. But if you look at the number of software curated here, there are more than 1,300 softwares. So this database is helpful if we're looking for some specific analysis tools, but maybe too extensive, especially if we are new to this field. There are some useful reviews here on some softwares, which will give you general ideas. And for example, this table from one of those review papers shows what's the widely used software, especially focused on open source softwares. So I would say when it comes down to analyzing images for your own research, and if you haven't done any image analysis before, I'd recommend you start with ImageJ or called Fiji, which is an ImageJ bundling software. ImageJ software, it's the same software, but it has many plugins included. And this is the most common software and give you access to hundreds of different plugins. Okay, now let's get to the image analysis workflow by starting on the basic rules to follow during microscope image acquisition. So acquiring proper images is really crucial in getting the proper measurement. And these are the 5 key rules you want to follow during acquisitions if you want successful image analysis. Some of them maybe slightly overlap our previous lecture, but I put it here because these are really important. And essentially, you will need to use appropriate resolutions and acquire with sufficient sampling rates, and also use proper bit depth, use whole dynamic range, and avoid saturations. So I'll go through one by one on the following slides. And to understand the basic requirements of acquisition that I mentioned before, first we need to know what microscopy image actually is. Well, let's say that these are the fluorescence signal of our objects. And when we use the microscope, this signal will be formed as an image through the optics. And this is currently an analog and continuous signal. But when this image is detected on our camera and saved on the computer, the signal has to be converted as a digital format. So the images we get is the digital image composed of many pixels. And pixel is the unit of our image, and each pixel has defined size. So when we convert our analog signal to digital formats, we need to use the correct sampling rates. Well, if we want to know what's the sufficient sampling rates, we can follow the Nyquist sampling theorem. So the Nyquist theorem is also known as a sampling theorem. It says that when you are capturing a signal, like an image, you need to sample it at a rate that's at least twice the highest frequency present in the signal. So what does that actually mean? As you can see in this image, the black curve is our signal, and the red dots is where we will be sampling. And when we sample this at its own one frequency, you can see that we cannot truly capture the information. Increasing the rate slightly higher improves it, but the signal is only start to be captured properly when we start to sample at least 2 times of its frequency. So in a simpler terms, if you want to accurately represent an object in a digital image, you must sample it frequently enough to capture all the details. So in microscopy image, the Nyquist conditions mean slightly different, and it means that the pixel size should be at least 2.3 times smaller than the object you're trying to resolve. So this factor 2.3 accounts for various factors such as optical diffractions and the shape of pixel also being square. So in other words, if you want to see fine details, make sure your pixels are small enough to capture them. So for example, let's say we want to resolve a cell with a diameter of 10 microns, then your pixel size should be at least 2.3 times smaller of these cells, which means around 4.3 microns or less. And this is an example image where the objects were, say, taken with ideal sampling rates. But instead of this Nyquist sampling, if we use lower sampling rates, which means larger pixels, then this result, as you can see, is too pixelated, and we will lose some detail of our objects. So there is some information loss happening. On the other hand, we can also use really higher sampling rates than Nyquist. But if we use too high sampling rates, this may generate prettier image, but there are practical drawbacks without necessarily improving our quantifications. For example, the imaging may take longer, and also file will be larger, which means slower in data processing in every step. So in summary, if we want what we want is the sufficient pixel resolutions in our image without necessarily oversampling. The things that actually should been set even before the sampling rates is choosing the correct objective lens to achieve correct resolutions, which means that if we use, say, low resolution objective lens, the analog image won't have enough information to start with. And if this happens, using the correct sampling rates will not actually restore the information. So similar to our undersampled image, this low resolution image will mislead your analysis. So we need to choose the appropriate resolution for our object, and how different objective lens gives different resolutions was, I believe it was covered in our lecture 1 in fluorescence microscopy lecture 1. So you can go back to this for more details. So I have mentioned that our microscopy image is a bunch of pixels, and these pixels have its own intensity value as well, as you can see here. And for example, this pixel on this image has an intensity value of 159, and its size is 32 nanometers in X and Y. But what this intensity actually means. So when we convert our analog signal to our digital image, we can also choose on how to convert the strength of signal into intensity values. So this can be converted simply as 0 and 1 in 1 bit depth image, or 4 gradient steps in 2 bit depth image, or 8 steps in 3 bits, or 16 bits in 4 bit depth. And at 8 bit depth, the signal can be addressed in 256 steps, and this gives smooth enough gradient for human eye, and that's why many microscope images is saved in 8-bit formats. So this may be good enough for visualizations, but actually for quantification, we can use higher bit depth, such as 12 bits or 16 bits, to get much more information on the signal. So the intensity will be recorded as 4096 grayscales in 12-bit image, and over 65,000 steps in 16-bit image. And as we use higher bit depth, we can detect the difference of intensity much better. So this example shows where the same analog signal was encoded in 8 bits and 16 bits. And in our eye, this 2 pixel intensity looks pretty much the same. And it is indeed the case with 8-bit image. But in 16-bit image, the encoding can be differentiated these subtle differences, which means that if you are planning to measure intensity, use higher bit depth than 8 bits if it's available on your acquisition system. Okay, let's have a look at another intensity feature of our microscope image. So this example image was encoded as 8 bits. Now you know what 8-bit means. So the intensity range is from 0 to 255. And if we plot how many pixels there are for each intensity value, for example, we can count 0, 0, 0, and 1, and 4, like this, for the entire image, then our image can be presented as a frequency histogram. And this histogram is very useful in checking the general intensity distribution of our image. So for example, this is the histogram of this image. And you can see that the majority of this low pixel intensity, in low intensity pixels, is representing the large background area. And the mid intensity and the bright parts are evenly spread out. Whereas in this example, you can see that image is much darker, and the total range in the histogram is skewed to the left. And in this case, we didn't even use the total 256 scales, and maybe perhaps utilizing only half of them. So the gradient coding is done only with this around 120 values. And of course, this will be less informative than the image with full dynamic range. Also, when our images is taken like this, the signal is not well separated from the background. So this will also result poor segmentation. So during our acquisitions, we have to pay attention to this histogram and try to use the whole dynamic range as much as possible. But sometimes we were trying too hard to make the images brighter and ending up saturating pixels like this. So saturation means that the signal strength reaches over the limits and the intensity value will simply encode it as the maximum value of your camera. And when this happens, we will never be able to retrieve the details. So this also should be avoided during acquisitions. And most acquisition system provides a visual range indicator, but the histogram is also very useful tools in checking this type of saturations. Just a couple of few other things to remember during acquisitions is that first, of course, the all images we are going to compare should be acquired and also processed using the same settings. And when you save the images, always save as a raw microscopy format or even OME TIFF to preserve metadata, which is very important in bioimage analysis. And we should avoid saving as a standard image format such as TIFF, JPEG, PNG. So, that was the first part of my talk, and I have covered 5 key rules for acquisitions. Next is let's do a pop quiz, see if anyone's been paying attention. So let me just start the first poll. Yes. Okay. So here I have put 2 simple questions. Both are single choice. So I'll give you about a minute and a half. Yeah, I'll give you 10, 15 more seconds. Okay, I'll close it now because we have most of people have participated. Just end the poll and I will share results. So for the first questions, what is the ideal size for microscopy? Ideal pixel size for microscopy? So it's good that most of people have got the correct answer. Yes, it has to be at least 2.3 times smaller than my objects, which is correct. And the second part is which bit depth should you use for intensity measurements? So here we have majority of people answered correctly 16 bit depth, but some people were also selecting the 8 bit depth. So as I mentioned, higher bit depths the better for separating those subtle difference of intensity. So it's always good to use a higher bit depths, whatever available on your acquisition system. So let me just stop sharing and move on to the next one. Does it move? Okay, there you go. So the next step to acquisition is pre-processing. So even if we have managed to take the best possible image from the microscope using those rules mentioned before, the quality of raw microscope image can still be compromised by manufacturers of acquisition systems, and we may still need to correct it by doing some pre-processings. So there are many different procedures out there. So covering everything will be impossible, but I will introduce 5 common correction procedures to give you some ideas. So the first thing to consider is denoising. Essentially, every microscope image contains a noise that are mainly coming from the electronics of the acquisition system. And this noise can be a problem in object detections, as you can see in this threshold image, highlighted in red. So there's many noises here. So the classic way to reduce noise is to capture more signals by increasing acquisition time and also sometimes average out those random fluctuation noise. But this may not be always possible, for example, when you're doing live cell imaging, so if you have image really quickly, or it cannot be also practical for acquiring large samples because it can take really long. So an alternative option is to reduce the noise using some image filters, such as median filter or Gaussian filter. So these are the 2 commonly used filters, where the median filter preserves the edges better, and Gaussian filter is to smooth the object, smooth out the shape of the object. So you can see that both filter makes object detection better, but slightly in different way. And most of image analysis software will have both filters, and there's so many more type of filters. So if you want to know more about different type of filters and how this filter works, this image processing handbook will be a very useful reference. And recently, there have been some deep learning approaches for denoising, and one of the good example is called noise to void. And if you're interested in this tool, you can check their own GitHub page. And there are also options where you can train your own data sets using Python or through a Fiji plugin, although it's a little bit complicated. Another important pre-processing step is the background corrections. So in many cases, you will find that the center of your imaging field is always brighter. And this makes it really hard to detect the object in the middle and the edge of the image using the same threshold. And this is due to the nature of the optics and sometimes due to the misalignments of the microscope. So it'll be best to remove these artifacts first by aligning your microscope before acquisitions. But even after you align it, there may be some residual defects. So we still need to correct such unevenness using correction algorithm. Some common correction tool is called rolling ball background subtraction. So this method essentially estimates the background as a large ball shape, and that's based on our user-defined value and subtract intensity. The other type of correction is called flat field corrections. And here we can also use an empty background image taken at the same microscope, and that was using exactly the same settings, and we can divide the raw image with the flat field image to get the corrected image. Sometimes your image may have some this type of stripe artifacts, and this type of things can be corrected using called bandpass filter. But there are also many other more advanced background correction tools, and one of them is called BASIC, which computes the shading matrix and corrects the shading. So as in this example, the shadings shown in large mosaic image can be corrected quite effectively. And this type of background correction tools is available in most image analysis programs, such as Fiji or Self Profiler or any other. And in fluorescence imaging, especially when you do some live cell imaging, your sample fluorescence signal may bleach over time, or sometimes when you don't have when the fluorescence lamps start to playing up, it may jitter over time or within your 3D stack. So this is an example where the image was taken over time and shown as a montage like this. And you can see the intensity of the sample bleaches over time. And this bleaching makes image analysis very difficult. So there are a few tools available to correct such bleaching issues. It's called bleach correction in Fiji or bleaching corrector in Huygens. And in this image, particularly, the decreased intensity by bleaching was fitted pretty well with exponential decay equations. So the image was successfully corrected by using the exponential fitting method. On the other examples, where the bleaching may not always fit exponentially, and then the image can be corrected by a method called histogram matching. But in this type of correction tools, we have to be very careful because this correction is useful for segmentations, but it's not really appropriate for intensity quantifications. So for example, if you want to quantify the intensity of this particular object, we can segment the object on the corrected image, but the intensity measurements will actually have to be done on the original image, as an example. The other type of correction tool is for correcting the misalignment. So image misalignments can occur in multidimensional data, and this can be in X, Y, or Z, or over time, and could be due to the drifting, where you can see in this type of example. So this drift needs to be corrected before any object detection or measurement, like this. And you can use tools such as correct 3D drift in Fiji. And there are many actually different drift correction tools available in Fiji as well. And also, this type of misalignments can also happen between different fluorescence channels, like this. So this is because of the chromatic aberrations. Chromatic aberrations essentially means that the different wavelengths of light is formed slightly on different plane or positions. And if you're looking at a very small object like this, you will notice such misalignments even with the calibrated lens. So this correction is especially important if you're going to measure colocalizations. So tools like Transform J or chromatic aberration corrector in SPI will help with this type of chromatic corrections. Another case that image needs to be aligned is when the microscope images were taken in multiple positions to create a big mosaic. So regardless of whether it's that your image is 2D or 3D, it should be stitched to make a seamless large image before any further analysis. So the Big Stitcher is a great Fiji plugin tool for stitching large tiles. And another correcting method is called deconvolutions. So deconvolution is a mathematical image restoration method where we can estimate how this light scatters on our microscope settings based on multiple parameters. And then we can restore the signal based on the calculation. So by doing this, we can increase the image resolution in X, Y, and Z. And as you can see in this example, the localization analysis of these two nucleus, for example, will be much more accurate after the convolution. So the deconvolution can also increase the contrast and remove noise, as you can see in this example. And altogether, the deconvolution improves the quality of data for visualization and analysis. The most commonly used commercial software is called Huygens, and it's available at our platform, so you can contact us if you'd like to access it. All right, so that was the short segments for the pre-processing. And let's do another quick quiz, pop quiz. Let me go back to start launch this. So here I only have one questions. And so I'll give you maybe 30 seconds. And this is a multi-choice questions. Okay, maybe 10 more seconds. Okay, I will, oh, still some more coming. And I'll finish the poll now and share the result. Okay, so this is what everyone has answered. So the question was, which features can be improved during pre-processing steps? The first one is signal to noise ratio. Yes, we can increase the signal to noise ratio. We can pretty much reduce noise using those noise reducing filters, Gaussian median filters. And also we can do this using deconvolution method. Not many people have selected the bit depth, which is correct. I mean, bit depth is kind of determined during the acquisition, so we can't really change it during the processing steps. And we can also increase the resolutions, like I mentioned briefly just before, as a part of the deconvolutions. So, but things that I didn't actually mention because it was too much details is that image has to be acquired a certain way so that we can process the deconvolutions to increase the resolution. So yes, the resolution is the right right answer, but it needs to be the image needs to be taken a certain way, very specific way. And unfortunately, we can't really correct the saturations. So again, during the acquisition, if the image was already saturated, there's no way to correct it. So that's why I mentioned that we have to be very careful in avoiding saturations during image taking. And yes, of course, we can correct the uneven background illuminations. There are a couple of different tools available. So yeah, thanks for answering all these questions. And let me move on. Okay. So the next workflow is detecting objects or region of interest for our measurements. So the object detection is usually done, majorly done in two steps called pixel segmentation or classification and binary processing. So the first step is to classify each pixel into signal or backgrounds category and reassign the signal to white and background to black. So as a result, we'll get a binary image like this. And getting object detection right is really crucial for accurate measurements. So there has been many different approaches developed. And I will introduce four main approaches in the next slides. And these include conventional segmentation, machine learning, deep learning, and some other computational methods. And once the pixel is segmented, most of the time we need additional tidy-up process, such as disconnecting connected components, or even generate the ultimate eroded points like this, which represent the center of particles depending on our needs. So these processes is usually done on our binarized image. So it's called binary processing. So let's have a look at the conventional segmentation process first. So if we want to segment the cells from the background, look in this image, we can have a look at this histogram of the image, and we can set the intensity value somewhere here as our threshold, because for me that seems like the right value to separate the signal from the background. Then we can generate a binary image. The thing is that during this step, other people may have a different idea and they may set the threshold to different value, and this will produce a very different result. And this inconsistency is especially a problem if you are measuring the area of the cells, for example. So what's the best way to find correct threshold value? One simple way is to set the threshold value using our negative control image, where you are not expecting any signal, or from the background area of the same image if you don't have any negative control image. And in this single threshold method, the important thing is to apply the same threshold value to all images in order to be objective. But in many cases, the real images have very different background levels, and even the same sample with the background level may change over time, especially when you're doing live cell imaging. So when image level background levels are so different, applying the single threshold value will not always work. So to compensate this issue, we can first use the auto threshold method. So in this method, the threshold will be calculated logically in each image. So there are many auto threshold algorithms available, as you can see here. For example, one of the methods published by Riddler is called isodata. And in this method, the threshold is found as a value where the average of the pixels above the threshold and below the threshold are identical. So you can see the equations here. Or another method published by Previtz is called intermode. And this method always assumes the histogram to be bimodal, as you can see in here. And it's trying to find the two local maxima here and there, and computes the midpoint of the two maxima. Well, this method may work well in this example histogram because it's quite nice bimodal, but it may not work if the histogram is broad and flat, or if the peaks are extremely unequal. So as one method may work better than the other method, depending on the type of image or type of histogram, what we need to do is we need to test which auto threshold method works best on our data sets. So for example, we can test up to 17 auto threshold method and 9 auto local threshold method in ImageJ and VG. And once we have found the best method for our image, like this, then we can apply the same auto threshold algorithm to all of our image. So in this case, it's slightly different to the single threshold method, where single threshold method is just using the single value, whereas in auto threshold algorithm, we apply the same algorithm. So the actual absolute value of the threshold may be different, but it will be calculated per image. but it will still try to find the background with using the same logical method. Well, the conventional thresholding approach that I've just shown you, it works well usually in fluorescence images and also when it has a good signal to noise ratio, but it doesn't work very well if the signal is not clearly separated from the backgrounds or if it's completely non-fluorescence image such as bright field, color, phase contrast, or EM images like this. So for this type of images, we can try machine learning approach. And in this approach, the pixel segmentation is done by user to annotate different pixel groups. So what that means is in most programs, you will need to label something like this, saying this pink is one group of pixel and this purple is another type of pixels like this. And it's usually done as line drawing tools, like a brushing tools. And once it's done and let the software to train, it will try to analyze many different features of the pixels, not just the intensity, but something like the color, texture, and the surrounding context. And then it will calculate what the likelihood of each pixel will fall into which category. So then it will generate the probability maps, which we can then convert into binary image. So most of the time, this type of manual annotation is quite simple, as in you don't really have to annotate the entire image. You just need to give some sample examples like this. So it's simple and interactive. You can usually see those results as you're drawing. So it sort of updates pretty quickly. And also the software is user-friendly. So the good news is that, especially for most of biologists, is that no programming skill is required. So we can easily import the image and do some drawings and get the result. So there are also many softwares available supporting this type of machine learning-based segmentation, such as Mares, Avia, Zen, IntelliSys in commercial fields, or QPath, Weka, LabKit, and Elastic, etc. for freeware. So you now have a lot of options to choose from. Another approach, automated approach to object segmentation is called deep learning. So for those of you who are not really familiar with the deep learning, let me give you some high-level overview. So the deep learning is a subset of machine learning, and that's the one that usually deals with the artificial neural network. So what is artificial neural network? So as the name suggests, it's modeled after the way that neuron works in the brain. So in general, the neural network can be thought as kind of black box like this, but it takes some input data and learn within, so it just creates many connections to try to learn and recognize some sort of patterns and structure of the data. And once the network has learned these patterns, we can use it to predict the outputs for new sets of similar data. So it's like input and output, but we have to train the model, essentially. And in image analysis, this deep learning method can automatically extract image features rather than manual way. So one of the deep learning method that's used a lot in image analysis is called convolutional neural network. So this is an example architecture. And here you can see that there's many layers of architecture. And each layer represents the sort of the convolved image that detects a certain feature from our input image. So for example, if our input was an image of a car, then in early layers, some basic features such as various edges and circles are learned in the model. But as we get deeper into the network, the feature gets increasing a bit complex, such as it can detect the wheels or door, or even higher to the entire car. So after we gives lots of input images like this and get the model to be trained, and the model can generate these complex feature maps, then it can aggregate all this information to make some sort of predictions about the input image. So that's how the deep learning is done. And in bioimage analysis, this, as I mentioned, it can be used to detect very complex patterns. So, for example, a deep learning model called UNet can segment tricky images like bright field or EM images. So this would be really hard to segment in conventional or sometimes even using the machine learning, those manually labeled segmentation. So whereas deep learning can perform really well for this type of image. Or another tool called STARDIST. It can detect specifically a nucleus type of round objects really well in both fluorescence and histology images. So this software, as you can see a bit more details, can even detect the overlapping nucleus. So it actually can represent a real nature of the nucleus much better than conventional method. Another really useful tool is called CellPose, and it can detect various type of cells and nucleus. So this is another one that used quite a lot. So here you can see even from those quick example that the usefulness of deep learning approach. The thing is, most of this approach requires a large amount of manually annotated data and also programming skills to develop the trained model. But luckily, there are some pre-trained models based on this type of images, which are accessible via user-friendly software such as Fiji, Cell Profiler, Elastic, and Elastic is through the bioimage model Zoo. So yeah, those programs can also be used to access those pre-trained deep learning models. And kind of the last one, which is called slightly different way of doing object segmentation in computational approach, is tool called template matching. So this method doesn't require any manual annotations, but it works in a way that you're providing either one or multiple template images, like you can see in this small inset, and also you provide a target image where you want to find a similar structure. So then the algorithm will calculate the correlation map between this template and target image and give you some candidates. So this method is particularly useful if you're looking for some specific structures that are similar but repeated in many images, such as time-lapse or tissue microarrays. And also the good news is that no programming skill is required to use this plugin. You can just install the plugin in your Fiji. Okay. So let's say we have segmented our objects based on one of those, actually 4 different methods. Then have the binary images, and then we can do further morphological operations. So for example, we can separate connected particles using watershed algorithm, or we can reduce the size using erode functions, which is removing the pixel from the outer layer one by one. Or we can do the opposite using dilate function. So it's adding a pixel, 1 pixel at a time from the object boundary. And if we need to do some more like tracing or different type of analysis, we can make the structure as a single pixel width shape, which what we call as a skeleton. And in some cases, we may need to fill holes if our object detection wasn't really perfect and you really want to measure the area of the whole cells rather than the one with the holes. Or we can also find the ultimate eroded points, as I mentioned before. Of course, there are many more processing, such as Euclidean distance map and Boolean logic operations. And all of these morphological operations in binary image is very basic processing tools like image filters. So it's available in most of image analysis softwares. And I will cover some of these processings and how they can be used in some later part of my talk. Okay, so that's already almost been an hour. So let me pause here and run the third part of the pop quiz. Again, just one question. I'll give you 1 minute. Okay, maybe 5 more seconds. Okay, I will close the poll and share the result. So in here, the question was, in conventional segmentation, what's the best way to set threshold on dataset with various background levels? So mentioned that if all images have slightly different background levels, first we can use auto threshold algorithm. So I think most of you have got to that correctly because I can see the answers are sort of divided exactly as the second and the fourth one. The difference between that is what we want is once we found the correct algorithm, we actually want to apply to just a single algorithm to all of our data sets. So we don't really want to apply different type of algorithm for each image. So maybe the answer was a bit not very clear. Essentially, what we want to do is use a single auto threshold algorithm for the entire data set. Hopefully that makes sense. That's the more objective way of setting the threshold. Okay, let me stop sharing. And okay, let's move on to the next one. I have to go a bit quick. Right. So now we are at the fourth stage of bioimage analysis, which is measurements. So these are the standard measurement categories, and I will introduce each briefly on the following slides. First measurement is number. So this is probably the most common quantifier in bioimage analysis. And when counting number, we can apply automated detection workflow so that we don't have to count these many objects manually. So automatic detection becomes more powerful when it comes to counting objects in 3D or even over time. As you can see, there's so many objects we need to count. So we definitely want to do it in an automatic way. And also in many cases, we will need to count object per discrete container, like it's a cell, for example. So this can be cells or any specific region of interest if you're doing any tissue section. So then the counting can be presented as per measurement area or volume or per ROI. The next common measurement for fluorescence image is intensity. And if we like to compare the brightness of the object in this image, we can measure the mean intensity of each object. So mean intensity is the sum of total intensity value divided by the area of object. So this represents the standardized intensity regardless the size of the object. So this is the right way to measure the brightness of an object. So let's say we have measured the mean intensity of each cells and plotted with beam plots like this. And from this plot, we can show that the treated cells are brighter, but also can show that there are two distinct populations in here. Apart from measuring the mean intensity, we can also measure other intensity features such as the minimum, maximum, median, sum, and standard deviations. And these quantifiers can be useful for measuring different features. So as an example, these two different objects have similar mean intensity, similar average intensity, but we can see that there's a clear difference that the left cell staining is smooth and the right cell shows more intensity fluctuations and structures are more granular. So if you look at the histogram of each image, the intensity range distribution is more widely spread on the right cell here. And we can measure such dispersions. So the standardized measure of it is called coefficient of variations, which is the ratio of the standard deviation to the mean. So this value can be the measurement for the degree of fragmentation or homogeneousness of intensity. And the next common measurement is the shape. So biological structure can be measured in many different parameters. Yes, the most common one is the space occupied by a shape, which is the area in 2D or volume in 3D image. Or we can measure the outline of the shape, which is called parameter or surface area in 3D image. Or we can also measure the spread of the shape using convex hull. So the convex hull is a minimal polyhedron that fits an object. It's like a shape when you put a rubber band around the object. And from this measurement, sometimes we can also present it as a ratio of two values to show the complexity of the shape. So for example, we can present it as a ratio of an area and parameter, or as a ratio of area to its convex hull area, or the ratio of parameter to its convex hull parameter like this. So as an example on how such ratio is used, for example, in this paper, the researchers have described the morphology changes of the cells in 3D using MRS. So when the glial cell here, shown here, is stimulated with ATP, which is one of the inflammation factor, the process of the cell becomes longer when cell was in the resting states. So you can see it's slightly elongated, but the stimulation actually retracted like this. It's shorter and thicker in LPS activate, if the cell was already activated with LPS. So you can sort of quantify this type of degree of cell ramifications using the ratio of the surface area to its own volume. You can see it's going up and this one is going down like this. If we want to know more about the size rather than area, we can also measure the width and height of the minimum bound rectangle, or even the maximum ferrite diameter, which is the longest possible line that can be drawn on the object, or aspect ratio of the minimum bound rectangle is a good indicator of the elongation or roundness, as I mentioned in the very first part of my talk. We are more interested in the branches of the cells, such as neurons or microglia, we can also trace them as a filament. And once done, we can measure the branch-specific parameters such as total length, level of branches, and angle of branches. And in this picture, the branches are color-coded by its connections. You can see their individual branches. And in this example, you can see sort of the orange line, which is the longest path that was detected in this particular filament. So we can do lots of more analysis once it's branched. And also with branches, we can measure the complexity of the branches using the shoal analysis. So this analysis essentially counts the number of branches per concentric shells. So it's count from the smaller circle as you go out, and as you start finding those number of branches, it will start count. So that was the shape, but in some applications, we also want to measure the relationship between objects. One way to do it is to measure the nearest neighbor distance between the centroid of the object. So this works well if the features are really small compared to the distance between them, or relatively round object. If the objects are large compared to its distance between them, and particularly when they are irregular in shape and vary in size, and also have a different orientations, anyways, complex, then it may be more appropriate to measure the distance from edges to edge rather than just simply based on the difference between the centroid. So here's an example where we want to measure the relationship between objects. So in this diagram, the left image represents a cell that, let's say, it's receiving the cells that are receiving synaptic inputs. And on the right image, we have a synaptic proteins staining on the whole tissue. So these are just schematic diagrams. But if we want to know how many synapses there are for these cells, and we will need to detect the particles that are touching the cells only. So we can do this by first generating a Euclidean distance map outside the cell. So this map is an image where the pixel intensity increases gradually from the border of the object. So then we can measure the intensity of this distance map for each particles. So essentially the particle with its minimum intensity 0 means its distance from the cell border is 0. So then based on that, we can filter the particles to select only the touching object. When it comes to the close relationship measurements, colocalization analysis is one of the most popular methods. But when we are referring the term colocalizations, we should be very careful not to interpret this as two molecules on the same location. So in bioimage analysis, colocalization analysis generally gives an indication that there are two fluorophores are generally near each other, and it doesn't indicate that the molecules are interacting. So when we measure the colocalizations, we can measure if there's any relationship between the channel intensities by looking at the correlation using Pearson's coefficients, which means, for example, if any brighter green tends to be associated with brighter red, something like that. Or we can measure if the fluorophore are generally near each other. So in measuring this co-occurrence, we can measure Mendel's coefficients, which measures the sum intensity of objects, or we can measure the overlapping area or volume, which is called based on the object-based method. So one also important things to note is that this type of quantifications is heavily dependent on the resolution of the acquisition system, as you can see in this example, that the low resolution image, the image was taken with NA of 0.25. Even if they are same sample, you can see the low res image, we can probably end up measuring colocalization much higher because the resolutions is low and each pixel end up containing more informations of the fields and they tends to overlap more. So then it can actually misrepresent the true nature of the sample. So we have to be planned very carefully. So the last but not the least measurement category is tracking. So when we have the time-lapse image like this, we can measure the parameters that are specific to tracking, such as speed, displacement, total distance, direction, straightness, or durations. So, for example, if we want to investigate the mobility of the cells, and displacement will be a good measurement. And you can see in this plot, you can see the reduction of the cell movement after direct treatment. Right. So this will be probably done one of the next fourth quiz. And here we have 2 questions. I'll give you a minute and a half. As the poll went through. Oh yeah, that's coming. First is single choice and the second question is multiple choice. Twenty more seconds. Okay, we have steady numbers. I'll just close the poll and share the result. So the first question was, what's the best quantifier for comparing brightness of microscope image? Most of them have got the correct answer, which is mean intensity. Yes, that's correct. And the second was rather tricky because I just covered it really quickly and expecting you to remember all of this, but most of you actually got it right. So the best correct description for colocalization analysis is, yes, it does measure the correlation of pixel intensity using Pearson's and also measure the general proximity of molecules or objects like Mendel's or overlap analysis. And as colocalization measurements on low resolution can be maybe misrepresent the data set. Yeah, so those 3 are correct, not the first one, which colocalization doesn't really measure the interaction of molecules. I'm just gonna stop share. Okay, let's move on to the last part of my talk. And I'll go through some of the example workflow to give you an idea on how the actual analysis works, and then discuss the last step of bioimage analysis workflow, which is reporting. So let me show you some example workflow for the most common quantifier in image analysis, which is nucleus or cell counting. So if we want to count the nucleus from the image shown here, we can actually approach in multiple ways. So one simple way to do this is by detecting the intensity maxima. So this can be done usually by first pre-processing the image using Gaussian filter to reduce some noise, and then running maxima detection. So you might have noticed that these measurements can be done even without any object detection, but it can be only used in certain way, where it only works if the structure has only one bright point per structure. It's a simple method, but can be used in only certain way. So rather common approach is to use conventional threshold for object detection. So first we can set the threshold. As you can see, the threshold signal is shown in red in this image. And based on that, we can generate a binary mask image, like a white image. And if there's some objects that are touching, we can also do binary processing called watershed to split the touching object. And then we can count those objects using connected component analysis, which is called analyze particle in ImageJ. So these are really conventional method. And another approach can be machine learning or deep learning, as I mentioned previously. So in Fiji, we can access the pre-trained deep learning models for fluorescence nuclei detection using the StarDisk plugin. So this model works well on this type of nucleus image. So, and also we don't need any pre-processing or binary processing in most cases. But each trained model will work properly only for the type of images that was trained on, as how that works for the deep learning, which means that we can use StarDisk for this type of image, but we cannot use the StarDisk for the other type of image. For example, if you want to detect the cells, then we'll have to find different trained model. So as you can see that even this simple nucleus counting, it can be done in multiple ways. And also, once we detect the object using analyze particle or the StarDisk method, each cell will be identified as an object, and we can easily measure the intensity or shape parameters. So another common workflow is counting positive cells. So in this example, we have two channels where the first channel shows a nucleus of individual cells, and the second channel shows a phosphorylated histone staining, which is a marker for detecting mitotic cells. And if we want to count the percentage of total mitotic cells from those two images, first we can detect individual object on each staining. And in doing that, we can also remove the object that are touching borders because they don't have the complete information. Then we can filter the nucleus object, which is total population, based on the pH positive object, which means if individual nucleus overlaps with this pH 3 object, we can count as 1, and if it doesn't overlap, we can count as 0. You can see the counted one is shown as 1 or white, or non-counted are black, 0. So this will give us the count for total populations and the positive populations. And we can plot in graph or even show in a proof image like this. Another common workflow is measuring the translocation of the molecules between cytoplasm and nucleus. So in this example, we have 4 images where we have the staining for nucleus and the protein of interest. Additionally, in here, we have the illumination correction function images like this. And these images are the illumination profile that was calculated from the microscope. So these correction functions can be either generated from the microscope software or image processing software. Then we can use those images to correct the unevenness of the background by applying division calculations. And this correction will help us to improve the object segmentations. We can then detect the nuclei from the nuclear staining image. And after that, we can identify the cells by using the nuclei as a seed region, then growing outwards until it's stopped by a neighbor. So once we have detected these two structures, we can then identify the cytoplasm by subtracting the nucleus object from the cell object. So we get like this cytoplasm mask. So once it's object detected, then we can measure the mean intensity of protein of interest of nuclei and cytoplasm area and calculate the ratio. So as you can see, most bioimage analysis workflow are done in multiple steps. And this is one of the published examples where the process is documented clearly as a flowchart. And some softwares like Cell Profiler provides environments where those type of workflow can be designed within the program. And in this program, you can easily add or delete individual analysis modules and make your own. Once then, the pipeline can be used for batch analysis. So, and also the workflow can be saved as a file, which then can be used as your own record keeping and also shared with other researchers. So on the Cell Profiler websites, they also provide templates of many example applications, and some of them are already shown in the previous pages. So, and these are really helpful understanding different type of workflow. So I recommend you go and check it out. And such pipeline environments is actually not available on other popular programs like VG or QPath. But on these programs, the workflow can be recorded through script recorders and applied for batch analysis. And if you're more interested in automating analysis, you can also try using other programs like IC or Python, MATLAB, and KNIME. There are a lot more. So it is good to have these many options, but at the same time, not every biologist needs to be a programmer. So if you like to automate your analysis or get further help on image analysis in general, you can of course first contact your local image analysis, or you can also collaborate with our platform. So the final step of bioimage analysis is reporting, and I'll try to be really quick. So when reporting our measurements, so reporting the details of analysis workflow is often neglected or done in a way that it's really difficult to reproduce. So most of us probably have experienced the case where we have found a paper that describes the method we like to use on our data sets, but really couldn't get it to work, mainly because there was not enough details. So in worst case, I even have seen some paper simply just mentions the image was analyzed in Fiji. So there's a lot of issue there. So to increase the reproducibility of bioimage analysis, many bioimage analysts around the world have got together and developed a complete checklist for publishing images or image analysis. So this was published in last year in Nature Method paper. And it says that, for example, if you're publishing a new analysis workflow, you should provide the detail of individual components used in the new workflow and describe the sequence and key settings as the minimal options. So you should also share the workflow and example dataset through online platforms such as GitHub. And it is also recommended to explain the rationale and the limitation of your workflow, and ideally provide the screen recordings or tutorials. So that would be really helpful for anyone who like to replicate your analysis. So if we include all of these informations, so this will help in establishing a new workflow within the community, and ultimately it can increase the impact of your work because other people will start to work on your workflow. So this paper also provides a checklist for what needs to be included in publishing images, not just image analysis workflow. So I recommend to read it through if you're going to, or if you already have been publishing microscopy images. So wrapping up this talk, these are some useful websites if you want to learn more about bioimage analysis. So first, our platform website contains materials from many image analysis workshops we ran in the past. So this site is regularly updated, including this lecture series and our upcoming video workshops. And Image SC Forum is the first place that I would go whenever I have some problems or questions. So it's a really active forum and contains very useful information. And of course, you can visit these websites that are specific for each software for more comprehensive manuals or examples.